Hello friends, this is Dr. Jyoti Saxena and I welcome you to Biotech Made Easy videos. Today's topic is virus, virion, viroid and all related terms explained. I think the most confusing terms in virology and microbiology are the virus related terms. So most of the students are confused here. So I'm sure that after going through this whole video, I'll make sure that all your confusions are over. So let's start the topic. We'll start with the viruses. Virus is a general term. It's a broader term also, and this is accepted in day-to-day vocabulary. Everybody is using it. So it's not necessary that you have to be the student of science or a student of biology to use this word. Anyone can use this word virus. Technically, virus is an acellular infectious agent. Acellular means it doesn't have any cell-like structure, doesn't have any membrane, doesn't have any nucleus or the organelles. And this is infectious because it can go into the host cell and there it can cause the infection. It is a biological entity that includes viral genome. The genome can be DNA or RNA. DNA can be double-stranded as well as single-stranded, and RNA can also be double-stranded or single-stranded depending upon which virus we are talking about. And then the genome is surrounded by a protein coat, which is a protective layer. Some of the viruses may have envelope also, and they produce enzymes, and they have a structure like DNA or RNA and surrounded by protein coat kind of a structure, and it has an organization also. For example, like it has different type of symmetry, radial symmetry or rod shape they may be, or spherical. So that kind of icosahedral symmetry they have. So that way these are the biological entities which have their own structure and organization. So they infect all type of living organisms. There are plant viruses which can infect plants, animal viruses which can infect animals. They can also infect the human. So different human viral flu. You must have heard about coronaviruses also, everyone is aware about. So, and there are also the bacteriophages which can attack bacteria. It can replicate when it is inside the host cell. That is the key feature of the virus, that it cannot multiply, it cannot reproduce until there is a host cell. So when it enters into a host cell, then only it can multiply. And the life cycle of virus starts with the attachment. So as soon as it gets a host cell, it gets attached to the host cell, then it enters into the host cell. There the uncoating takes place. That means the protein coat is removed and the genome remains there in the cell. Then the replication takes place, and transcription and translation also takes place. The proteins are formed and the protein coat is formed separately and the genome is multiplied separately, and then the assembly takes place. And at last the viruses, the progeny of the viruses is released from the cell. So virus is the term used only for the active virus particles. So this you have to remember all the time, that when the viruses are in their active stage, they are, and we all know that they are active only when they are inside the living cell. They must need a host cell. There they are multiplying, they are showing the metabolism, they are making the enzymes, they can capture the host cell, they are multiplying there. So this active stage, when they are completing their life cycle there, so that period is the one where the term virus can be used. So not only disease causing viruses are there, because most of the time we think that viruses can cause different type of diseases, which is true also. But then there are many beneficial viruses also. So this is, different type of viruses. Here you can see some of the first one, the yellow one is the, rod shaped virus, TMV, and then the others, you may see the spikes and all. So different type of viruses are there. Then the second term is virion, And the most confusion is among the virus and virion. So let's see how. So it is a complete infectious particle present outside the host cell. So if you see the virion, then it will look exactly like virus. So it is complete infectious particle. It has a potential to infect the host plant also. But virion is the term which is used when the virus is present outside the host cell. So only when it is outside of the host cell, when it is not in its active state, then this is known as virion. And a virion is a virus in its extracellular form. That means when the cell or host cell is not present, it is in extracellular form, then this virus can be called as the virion. And it has all the components of the virus. For example, the genetic material is there, DNA, RNA, then the protein coat is also there. If the enveloped virus is this, then the envelope is also present. So all the things are, intact. They are just like virus. It represents a transition phase from one host to another. So what does that mean? That means when the virus is present inside the host, it multiplies and ultimately it is released from the host cell. From that point, when it comes outside the host cell and it comes into the environment, then you start calling it as the virion. And because it has a potential to infect the cell, so this transition phase, when it is present in the environment, in air, water or soil, wherever, and then it is waiting for another host where it can enter and it can cause again the infection. So that transition phase is known as virion. And it is inactive during this time. It is in dormant phase, although it has potential, but it is not entering into the host cell until it finds a host cell actually. So this inert phase is known as virion. So that is the basic difference. When it is inside the host, you call it virus. When it is outside the host, you call it virion. So the key difference is inside the host, when it is replicating also, it is multiplying, it is completing its life cycle, you call it virus. Outside the host cell, when it is inert, it is dormant, you call it virion. So I think you understand now what is the difference between virus and virion. Then the next term is viroids. Let's see, what are viroids? These are one of the subviral particles which are smaller and simpler. When we do the classification of the viruses, for a long time it was understood that viruses are the smallest and simplest organism present on the planet. But then afterwards the viroids were discovered and some other particles were discovered, and which were even smaller than the viruses. So they are called as subviral particles. So viroid is one of the subviral particles, very small, very simple. These are single-stranded circular RNAs. Only RNA is there, that is one thing. And this is single-stranded RNA only. And this is circular one and very small. How small? 250 to 400 nucleotide long. Very simple circular RNA is there. And unlike viruses, they don't have any protein coat. If they don't have any protein coat, there is no question of envelope. So no protein coat, no envelope, no formation of protein or enzyme. So very simple, just RNA particles are there. So this is the diagram of RNA, single-stranded RNA. Here you can see the single-stranded circular RNA. But then this circular RNA, it infolds. That means the base pairing takes place. And so this kind of rod-like structure is formed. And this is actually the viroid. Nothing else, but this is viroid, where the base pairing has taken place and the long rod-like structure is formed. So these two are the different type of structures which are formed. Then, hence depend heavily on the host constituents for replication and pathogenesis, because it doesn't have anything, just the single-stranded RNA which has enfolded. So if it has to be replicated, then it has to get all the components from the host. So very heavily it depends on the host constituents for replication and also for the infection. Replication is by rolling circle mechanism. This is one of the mechanism of the replication. So this one is here in the viroids. And these are infectious in nature, of course. They can enter into the host cell, there they multiply, and they can cause many diseases in plants, especially in the flowering plants. And potato spindle tuber viroid, this was the first viroid which was identified in potato. And here in this case, here you can see the potatoes which are infected by the viroid, and they become cylindrical. So they lose the shape, their original shape. So this is potato spindle tuber viroid present on the potato. So this kind of a structure is in viroid. Now there is another term which is known as virusoid. Now let's see what are these virusoids. This is also subviral particle. That means they are very small and they have a simple structure. So let's see how they are different from the other subviral particles. This also has, circular single-stranded RNA molecule, just like the previous one. So same structure, circular single-stranded RNA molecule, and also very simple structure and also very small. Almost the size is also same, 200 to 400 nucleotide long. So all these three characters, they are just like the viroid. But unlike the viroids, they cannot replicate on their own. You saw that viroids were replicating by their own, although they were dependent on the host components, but they were not needing any other helper virus. But here, they cannot replicate on their own. So how do they multiply? They require a helper virus to complete their life cycle. That means if the helper virus is there, only then they can complete their life cycle, otherwise they cannot. They are encapsulated by a helper virus coat protein. So this is another major difference between the virusoids and viroid, that here these virusoids have to be encapsulated. Encapsulated means the genome, the RNA, it has a viral coat protein. So in viroid there was no coat protein, and it was just like that, naked RNA molecule was that. And that was why it survived as naked RNA. It was because it made a very stable structure, because it was enfolding. So very stable structure it formed, and in that stable condition it can remain there in the environment, and it can also be taken up by the plant cell as a naked RNA. So these are the qualities which are present in the viroid, but here the encapsulation is needed. So protein coat formation is needed, and that is done by the helper virus coat protein. They infect a plant, of course, just like the viroid. They also infect a plant, but then they can infect the plant with the help of helper virus. Naked RNA cannot enter. In viroid, the naked RNA was entering, but here the helper virus is needed. So that is the basic difference between the virusoid and viroid. Then the third subviral particle is the satellite viruses. Now let's see what are these satellite viruses. These are again small viruses because it is subviral category, and they may have DNA or RNA. The viroid and virusoid, they had only RNA, but it can have DNA and RNA, and they can encode their own protein coat also. So that way they are different. They depend upon a helper virus for replication and infection cycle because they lack essential genes. Although they have DNA, RNA, but they do not have those genes so that they can do the replication and infection cycle by their own. So they also need the helper virus. And they co-infect a host cell with a helper virus. Suppose they reach to a host cell, so then a helper virus should also be there. So they co-infect the host cell. They are also there. Then the satellite virus is also there, and the helper virus is also there. Then they will do the infection. And when they are present along with the helper virus, then they can alter or modify, and most of the time they worsen the symptoms of helper virus. Helper virus is also infectious. It is also causing certain symptoms, certain diseases in the host. But if this satellite virus is also there, then it will, aggravate the situation. The disease will be much more obvious. And they can infect both plants, animals, human. So they can infect plants also, they can infect animals also. And the examples are satellite tobacco mosaic virus. This is a satellite virus and it needs TMV as a helper virus. Second example is satellite tobacco necrosis virus, and it needs tobacco necrosis virus as a helper virus. So you see here in this case, helper virus is needed. It can have the DNA or RNA. It has its own protein code, but it always enters into the host along with the helper virus. Now the prions. This is very unusual and very strange type of infectious agent. This is also infectious, but you see how different it is. They are just misfolded version of the normal cellular proteins which are found in the brain. See, in brain there are many type of proteins are there, and the protein folding is something which is very common. But here what happens that some of the proteins, they are misfolded. They are folded in abnormal way. And these abnormal proteins, because of the misfolding, these are known as prions. And here no DNA and RNA is present. So only the protein is there, but no DNA and RNA. And these proteins, they have this unique feature that once this misfolded version of normal protein is formed, they also trigger the other normal proteins to form the prions. So they are also misfolded, and then they accumulate together. And when they accumulate, then they start, damaging the brain or disrupting the function of the brain. So, and then, some abnormalities start in the brain. They cause neurodegenerative diseases due to accumulation of misfolded proteins, and the symptoms are like memory loss, confusion, movement problems. So all these problems take place when the prions are there. And they are extremely resistant to heat, chemicals, UV, even by autoclaving also. They are resistant. They cannot be killed. So very unique group. Then the last one is virus-like particles. Now you see why we are calling it virus-like particle. These are non-infectious. So first thing is that these are non-infectious. Until now, whatever terms we were using, they were all infectious. All infectious agents, but this is the only one which is non-infectious particle. Then why you are calling it virus-like particle? Because it looks exactly like real virus. It mimics like virus. They don't have DNA, RNA inside. So then what do they have? They have only the protein coat structure, which looks exactly like virus, but it cannot replicate, it cannot infect, or it cannot cause the diseases because it doesn't have genome, it doesn't have DNA, RNA, only the protein coat is there. So they are made up of viral structural proteins which self-assemble. So if all structural virus proteins are present, then they can self-assemble and they form a particle-like a structure which look exactly like virus. So that's why we call it virus-like particle, but it is not virus. So these viruses are not real viruses, they just mimic like virus, and this quality is used by the scientists and the industrialists for making the vaccines and for drug delivery. How? Because when they are present inside the body, when they are injected into the body, our immune system thinks that this is a real virus, and then all the immune response starts. So, but although it is looking like virus and our immune system is confused and it is giving the response, but actually it cannot infect because it doesn't have any DNA RNA, so it cannot infect. It is completely safe. So that's why it can be used as vaccine and drug delivery. So that is the main purpose of virus-like particles. So these can be prepared by the scientist, and when the structural proteins are there, they will self-assemble, they will make a virus-like a structure, and then these virus-like particles, these are used for the purpose of vaccine and drug delivery. So they can be very helpful in making the vaccines and all. The examples are human papilloma virus, HPV, or hepatitis virus. So these are, these virus-like particles are used. Now the summary is like when we say just one-liner, virus is entire infectious agent inside the host. So always remember, when it is inside the host and when it is entire infectious organism agent, then you call it virus. Then virion is the infectious particle, but when it is outside the host. So the key point here is that it should be outside the host and it should be in the dormant stage. Viroid is the infectious naked RNA. It is just naked RNA, but this naked RNA is very stable and it is taken up by the plants and it can infect only plants. Then virusoid, it is a naked RNA, same as the viroid, that needs helper virus to multiply, and encapsidation. So these are two things. One thing is that it needs a helper virus, and the second thing is in this case the encapsidation also takes place. That means it has to be covered by the protein coat. Then satellite virus. Satellite virus is the one which also depends on the helper virus, and it has DNA, RNA both. Then prions. Prions are totally different. These are infectious proteins which are present in the brain. And virus-like particles, these are the empty viral shells, no DNA, RNA, and this is used for vaccines and drug delivery. So that's how these different terms, which are all related to the viruses, but they are not actually virus, they are a little different. So I'm sure that now you do not have any confusion, but still if you have any confusion, you can ask me in comment section. So that's all for this video, and if you like the content of the video and it was helpful to you, then please subscribe my channel, and if you want something else, the videos on some other topic, then you can tell me. And thank you so much.